Start with the chapter 4 of plant nutrition, which is syllabus 5090, and this is the second video on this chapter. And basically what we're going to study is the rate of photosynthesis, the three investigations that were left from the first chapter. And now the syllabus point which we're going to cover now here is the syllabus point D. Investigate and state the effect of varying light intensity. First varying light intensity, then varying the carbon dioxide concentration, and then varying the temperature. And what we have to study is the rate of photosynthesis. Now, previously what we studied was necessity for chlorophyll, light, and carbon dioxide. So these were three practicals which we did in the first video, and now we're going to do three practicals in the second video, which we're going to be examining the syllabus point 10. The first thing is we just go through the structure of the leaf. It's got a waxy cuticle, waterproof layer, then there are veins, and there's the midrib. The midribs contain the xylem and the phloem, then the stem, and then the stem, then there's this leaf stalk or the petiole which comes out, and there's the main midrib which has a lot of it's quite thick, and it has the phloem and the xylem. Then the stomata are the pores on the underside, and this is where the carbon dioxide is going to enter and the oxygen is going to diffuse out on a bright sunny day. So this is just a recap of the structure of the leaf, then a recap of what we did, destarching the plants. How did we do this? Why did we do this? We had to keep the plant away for 48 hours, converted glucose for growth and energy. The plants will not contain any starch and are ready for the investigation. And then when we set up the experiment, we find out. Now this is just a recap of the previous video. Then how is the plant destarched, leaving it in the dark for 2 to 3 days? And what happens to the starch used for respiration? Then we have the 4 steps in testing a leaf for starch. You dip the leaf in boiling water for 30 seconds. Then you boil the leaf in alcohol. And you dip the leaf in boiling water. And then, of course, this boiling water is not necessary. It could just be water. And then you spread the leaf on a white tile and place the iodine solution on the leaf. Now, this is just the 4 steps in testing a leaf for starch. Now, what do we have to study? We have to study the rate of photosynthesis. And the rate of photosynthesis, we have to study the effect of varying light intensities. The word light intensities, do not say light only. You write just light in your paper and I can't give you a mark. So varying the CO2 concentration and varying the temperature, and we have to study the rate of photosynthesis. Now, in order to study the rate of photosynthesis, I want you to look back again on the equation for photosynthesis. So we have carbon dioxide, we have plus water, and what do we get? We get a glucose molecule, C6H12O6, and plus we get oxygen. Now, how can we study the rate of photosynthesis? Either we can find out the rate of CO2 uptake, how quickly, how much CO2 is taken up. Like if you have a closed room in which the carbon dioxide molecules were 1,000, and then in 5 minutes they went down to 500. So you know 500 molecules of carbon dioxide were taken up for the process of photosynthesis. So either we study the rate of uptake of carbon dioxide, or we can study the reduction of oxygen produced. Please remember, rate is always got to be something with time. So the oxygen produced per unit time, how much oxygen is being produced per unit time? Is it 10? Is it 20? Is it 30? Is it 50? Is it 100 per second or per minute? So the volume of oxygen produced per unit time. So we can study that, and that would give us the rate of photosynthesis. Now, why aren't we studying the formation of glucose? The reason is that, you see, glucose is not doesn't remain as glucose. It is immediately converted to starch. So can we somehow study the rate of starch production? No, we can't. At times we can, at times we can't. Because you see, that is why we de-starch the plant before, so that we would know how much starch is being produced. Is it 10? Is it 20? Is it 30? Is it 100? But that's a little difficult to do. So we do talk about it, but we do not actually do any experiment to study the rate of starch formation, and that would give us the rate of photosynthesis. Now, the simplest of experiments, that the simplest of apparatus that we can use is that we have a beaker, and in that we have an inverted funnel, and in the inverted funnel we put a little bit of plasticine here and we fix this up, and what we have is a pond weed. Pond weed would be an aquatic plant, because your syllabus also says use an aquatic plant. And then we place a lamp, and this lamp is a bit crooked, yes, I agree with you. And we place this lamp, it's not a very pretty lamp, and this lamp has a 100-watt bulb, and then we place in front of it is a heat shield. A heat shield is two sheets of glass, and it's got water in it. So what is going to happen is light is going to pass, but no heat, because it's going to prevent any heat, because the lamp would also emit a bit of heat, and this could then affect the temperature of the plant. So we don't want the temperature to be affected. So we place this heat shield in front of the lamp, which is going to allow light to pass, but it will not allow the temperature to change, because even a slight bit of heat could affect the temperature of this pond wheat. So number one, lamp with a fixed wattage of the bulb, then the heat shield, and then we have this beaker, and in the beaker we have the pond weed. But the trouble is, now what are we going to add to the beaker? What are we going to add is we're going to add some sort of a liquid to it. Now the options are what? Please remember, it will be all over. This funnel is not going to restrict, the water is going to be all around it. So this is, we have the options here. You could have just put tap water or distilled water. No, well, we don't want to do that, because you remember the pond weed must be in a pond. In a pond, there must be frogs, there might be tadpoles, there might be fish, and these are all producing carbon dioxide. So what we have to understand is that we'd have to put some sort of a chemical in it. So you could, well, I think we could put 2% sodium hydrogen carbonate solution. And this sodium hydrogen carbonate solution is going to provide the CO2. Is going to provide the CO2 to this plant. The plant needs CO2. And after some time, what do we see? We see some bubbles coming out. And these bubbles are of oxygen. And we can count the bubbles per minute. We can count the bubbles per minute. So we count the bubbles per minute, and we have, say, 10 bubbles per minute, and then we have 12 bubbles per minute, and then we have 20 bubbles per minute. Now something is wrong. Either this is wrong, or this is wrong, or this is wrong. Because you see, you have maintained a certain temperature, the lamp is at a fixed distance, and now we're having, say, 10 bubbles per minute. 10 bubbles per minute. And then you do another reading and you find it's 12 bubbles per minute, and then you find another one is 20 bubbles per minute. Now, if the next one is 11 bubbles per minute, you know this one is wrong. And this is called an anomalous reading. Anomalous reading. That means there is some anomaly. Anomaly means there's some sort of mistake. Because please remember, these are enzyme controlled reactions, and they would have to follow a set pattern. It can't be that suddenly the enzymes are going fast forward and the enzymes are working in slow motion. No, that's not possible. So what has to happen is that it has to be, say, 10 bubbles per minute or maybe 12 bubbles per minute, and maybe now after this there could be again 12 bubbles per minute, could be the same. And please remember, one of your MCQ checks this, they can't be 12.5 bubbles. You can't have half a bubble. Now, of course, this is very, very scientifically incorrect because bubbles can vary in size. So another better bet would be that we place some sort of a gas syringe on it, and then we can place this gas syringe on it, and then this oxygen is going to collect in this gas syringe, and then we can measure the volume of the gas syringe. So that would be an improvement on this experiment, is that we have a gas syringe instead of counting the bubbles per minute. Another experiment which has been set up, and you can see that this has a bulb and this has a dimmer switch. This has a glass heat shield, and then we have the Elodia, which is the pond, it's an aquatic plant, and you have the support for the funnel. Please look at each point, and then you have the bubbles, and you have the bubbles collected in this at the top. Now, why do we have a dimmer switch? The dimmer switch is because we want to vary. Now it says varying light intensities. See, if you didn't have a dimmer switch, well, you could move the lamp. But here you have a dimmer switch, so you can increase or decrease the light intensity. Increase or decrease the light intensity. So this is going to help us in studying the gas collected. So if we know the volume of oxygen collected, now if that volume of oxygen collected is going to change by changing the intensity of the light, then we can plot a graph and see the effect of that. The other experimental methods that we study the rate of photosynthesis. This is a rather simple one. And again, gas is being collected, pond weed, water, oxygen bubbles. Here again, we have bright light, and here we have water of sodium hydrogen carbonate, filter, funnel, beaker, inverted test tube, simple experiment. Here, there's a slightly different one here, tank or beaker water. So just giving you an example of how we could set up these apparatuses, slightly changing them, but basically there is one common thing. The common thing is there has to be a light source, and here we have to have pond weed, which is in a watery environment, and the watery environment we add a chemical which is sodium hydrogen carbonate, which is going to release carbon dioxide. Now coming back to this diagram, varying light intensity, as we said, either we move the lamp or we can have a dimmer switch. But now what I want to talk about is how are we going to vary the carbon dioxide concentration? How are we going to vary the carbon dioxide concentration? Now, the first thing that we're going to do is we're going to keep the lamp in the same place. The lamp stays there. The heat shield stays there. What we're going to change is this. Now, what are we going to change? We're going to first use 2% hydrogen carbonate solution. Then we're going to use 4% hydrogen carbonate solution. Then we're going to use 6% hydrogen carbonate solution. But please remember, important point, the volume must remain the same. Volume must remain the same. So say you added 40 ml of the sodium hydrogen carbonate. Well, then each time that you do the experiment, it should be 40 ml or 50 ml, whatever you want to take. But what you're going to change is the concentration. So first you use 2% sodium hydrogen carbonate solution. You did the experiment. Then you took out the pond weed, you washed it. Then you put it back into a 4% sodium hydrogen carbonate solution. Then you took it out, you washed it, and then you put it in a 6% sodium hydrogen carbonate solution. Please remember, whenever you're using a liquid, you've got to specify the volume. You have to tell me what was the volume of the fluid that you used. In this, of course, it was sodium hydrogen carbonate solution. So you're going to change the concentrations and you're going to study the effect of changing them. How does it affect the rate of photosynthesis? The third one that we had to study was the effect of temperature. So how are we going to study the effect of temperature on the rate of photosynthesis is that we're going to place this in a water bath, and we're going to have water in it, which is going to have it, and we're going to have some sort of a hot water in it, and or it can be a thermostatically controlled water bath, and we're going to change the temperature. So we're going to have the temperature maybe say 35 degrees Celsius, then we're going to study it at 40 degrees Celsius, then 45 degrees Celsius, and then 50 degrees Celsius. So we're going to study the effect of temperature on the rate of photosynthesis. So we're going to change it by using a water bath and keeping the water bath at different temperatures. And we usually take 5 temperatures to study the effect of temperature on the water bath. And we must take a varying number of temperatures because that is going to make you understand whether temperature, what is the different range of temperatures, because you have to realize every plant would have its own optimum. And then if you exceed the optimum, only then the rate of photosynthesis would drop. So you need to know how we're going to study the effect of, just a recap: rate of photosynthesis, varying light intensity, varying carbon dioxide concentration, and varying the temperature. How does it affect the rate of photosynthesis? Now, the next topic is the lack of nitrates on the growth of plants. Now, what we need to understand is that a leaf, when it photosynthesizes, it can make its own glucose. But what it needs is nitrates from the soil. And if it doesn't get these nitrates from the soil, well, what can happen is that the nitrates is added to the glucose molecule, and this results in the formation of an amino acid. And then these amino acids are converted to proteins. So if a plant has less nitrates available in the soil, the nitrates have to come from the soil. So if the plant has less nitrates, then the plant is going to be, we say, stunted growth. Why? Because nitrates are needed to make amino acids, and amino acids are needed to make protein. Also, nitrogen is also needed for DNA, for RNA, and for the ATP molecule. So nitrates are needed for the nitrogen part of the nitrates are needed for lot of very basic molecules which are needed for making a new cell. Just like when you want to make a new cell in a plant, you'd need to make a new cell wall, you'd need to make a new cell membrane, you need to make enzymes, you need to make the nuclear membrane. So all those require, of course, fats. Membranes all require fats, but then in the nucleus there is DNA chromosomes are made up of DNA. So if you need nitrogen for the DNA and you need to add new cells where you need more of the nitrogen, which is the form of nitrates from the soil, please remember the plant cannot take nitrogen from the air and use it. This is the lack of magnesium. You see, like magnesium also is obtained from the soil. So if there is less magnesium in the soil, magnesium basically is needed to make the chlorophyll molecule. The chlorophyll molecule is present in the chloroplasts. And the chloroplasts are present in, if you remember, we just did a recap of it. Chloroplasts are present in the palisade mesophyll, in the spongy mesophyll, and in the guard cells. So the chlorophyll molecule is needed for photosynthesis to trap light energy. And if the light energy is trapped, only then it is converted into the chemical energy, which is glucose molecule. So now what we need to understand is that if a plant has less magnesium available to it from the soil, it is going to have less chlorophyll. Now, if it is going to have less chlorophyll, then what is going to happen? Less chlorophyll means what? Less photosynthesis. Less photosynthesis means less glucose. So less photosynthesis, less glucose. Now, if there is less glucose, that means less respiration. That means less energy released. So less respiration, less energy released, and this results in less growth. So less energy released, and this will be less growth. So please understand this is that if we're going to again say this is going to result in stunted growth, but the main thing is how are we going to prove this? We're going to prove this by an experiment. You're going to take two large boiling tubes and you're going to place 2% nitrate solution in A and a 5% nitrate solution in B. You're going to have the same plant in each, and you're going to have the plant, you have the roots immersed in it. These are the roots, and you fill this up with the 2% nitrate solution. Similarly, you fill this up with the 5% nitrate solution, and you leave these for at least 2 to 3 weeks. Please remember, you cannot measure growth in hours. And you let these plants and you keep them, you give the light provided to them is say 8 hours every day, because you're doing that in the lab, and you're providing it the air and the carbon dioxide, and you're keeping them at a fixed temperature. So the temperature is fixed as the room is air conditioned, and the temperature is the same for the next 2 to 3 weeks, and then you let the plant grow. Now, after 2 to 3 weeks, what you do is you have to calculate the dry mass. You have to take out these plants and calculate the dry mass of the plant A and plant B, plant A and plant B. Now, naturally, the one that which has this one would have grown much more, because you see, maybe the number of leaves are the same. But what does the dry mass tell you? Dry mass tell you the exactly the organic matter. How many cell walls did it have? How many cell membranes did it have? And why am I saying dry mass is because I'm removing the water from it. Because the water can be variable. It can be 70% water in one plant, maybe 75% water in the other plant. But when we're calculating the dry mass of plant A and plant B, well, we actually finding out that was there a difference in the growth? Was there more growth here? And was there less growth here in A? Now, if there was less growth here, only then we can say this has resulted less nitrates. It says lack of nitrates. So less nitrates and less growth. So we say that is why this plant it has stunted growth. So please understand this phenomena of stunted growth and how lack of nitrates results in less nitrate available, less amino acids and less proteins. So the growth is not going to be the same as that in which a plant has been given more nitrates. When we come to the topic which was not covered in the previous video as well, why is it that we say that most life forms are dependent on photosynthesis? We don't say all life forms, but we say most life forms. So there are some life forms which actually do not need light, and they are from chemical to chemical. So chemosynthetic bacteria, they can actually use a chemical and get energy from that. But why do we say most life forms? Because if you look at all the food chains that we have, all the food chains that we have, basic starts from producer. Now, producer has to be an autotroph. Autotroph means that which makes its own food. So autotrophs convert light energy. They convert light energy to chemical energy. So autotrophs, so that is why if producers are all autotrophs, and then after the producer, then we have primary consumer. Like when you're eating an apple, you're a primary consumer. And then we have, of course, the secondary consumer. So the secondary consumer would be when you're eating a piece of chicken, because the chicken ate grain and now you're eating the piece of chicken. So why most life forms are dependent on photosynthesis is because Producers are the first, it is called the first trophic level. Producer is called the first trophic level, the first feeding level. So if it's called the first feeding level, it means that producers can only make food if they can photosynthesize. So most forms of life are dependent on photosynthesis. And this is what we understand in biology is because all food chains start with the producer, and producers are always plants, which can make their own food. That covers nearly all the topics which have been prescribed in the syllabus, and I'll just go through the syllabus once again so that we can recap. If there's anything left, we can just do that again. Please always remember to go through the syllabus once you have read that chapter so that you can check whether you know each of the points. Understand that photosynthesis is the fundamental processes by which plants manufacture carbohydrates from raw materials. Then we have investigate the necessity for chlorophyll, light, and carbon dioxide. Then the equation in words or symbols. Then investigate the effect of varying light intensity on the rate of photosynthesis. And then if there's submerged aquatic plant, we just did that in this video. Understand the concept of limiting factors, and describe the intake of carbon dioxide and water by plants. Understand that chlorophyll traps light energy, converts into chemical energy. Explain why most life forms are dependent on photosynthesis, then identify and label the cuticle, and understand the effect of a lack of nitrate and magnesium on plant growth. So this covers mostly all the topics which we have done in the syllabus. Please go through the book, whichever book suits you, and then see what are the questions. Please do the exam past paper questions, get the topicals, and do the past paper questions, do the MCQs and the paper 2 questions, and I think you should be good to go. Thank you.